Good afternoon, Dr. Joe. How are you?
I'm doing well today. It's an exciting day.
Thank you for making the time to talk to us in a few minutes. But also, we've had exciting 1.5 days of being in the Pulse Printed by Conference, which is the first of its kind, hosted by onecelldx Mumbai. What has been your experience? How you like the conference?
The conference has really been fantastic. Really diverse opinions. Very informational. Really a group of enthusiastic clinicians looking toward the future, looking how to provide the best possible care for their patients, and really looking at how they advance precision medicine.
That's great. And so what we want to do today is very quickly get like snapshot of the journey. You share the best in a very chichi he or she's out. And how do you decide to do what you did from the failure? And then we'll kind of let question that.
Yeah, so I have what I would consider an interesting journey, probably not one that's matched by too many people. I wound up in college playing baseball and broke my wrist after one of the seasons, and had my right hand in a cast for 13 months. So I couldn't write. And so to get around that, I happened to go to a small college in Iowa called Grinnell College, which was endowed by Robert Noyce, who worked for Intel and invented the integrated circuit. And so they had these very flat keyboards present. And because I couldn't write and I had to type and I couldn't use a classic typewriter, only a flat keyboard, I learned everything that there was to know about computers and wound up writing my own word processor and printer drivers and things to survive. And so the significance of that journey was when I got to medical school, I met the chairman of the pathology department who had, in his mind, rightly so, that computers and medicine needed to go together. He wasn't quite sure how, but he understood the importance of that. And he sort of took me under his wing, even as a medical student, and said, you need to go into pathology. And so that's actually how I got into pathology. It wasn't a love of pathology. It was mentorship from somebody that aligned my skill set to what he saw as the needs in medicine. And so that's what really set the journey on its pace. So, my first introduction was a problem. He was running an NCI program for bladder cancer, and they were having a major problem in discrepancies between reads between pathologists, and they had no way of resolving that without getting the pathologists together and getting them, in one room and making a decision. But they wanted to enroll patients in clinical trials in real time, and they couldn't figure out how they would do that. And so Ron Weinstein, who was the chairman, presented that problem to me, and I invented the robotic microscope with satellite imaging at the time because the internet wasn't available. And so that's really what got me into telepathology and the concept of digital pathology and expert systems and how we could standardize diagnoses, et cetera.
Wow, that sounds like quite a journey, and that's bigger. It's not too people, a handful who have that kind of.
Exactly. So it gets crazier. So I was a breast pathologist predominantly. And so the phase 2 trial of Herceptin was run at my institution, Rush University in Chicago. And so I was charged with looking at HER2 before anybody knew what HER2 was. And so I was looking at it both immunohistochemically. I brought up the FISH assay because Genentech was saying that was going to be important. And so brought that up and got, a world of experience before anybody else really knew about that. And so that afforded me the opportunity to talk to the scientists at Genentech and other thought leaders about really the first precision medicine drug. And so what I recognized from there was if precision medicine was going to work, there was no way that testing could execute throughout the United States the way we were currently organized, because we could barely do it at the university, and 90% of the cancer care is done in the community. And so I started a reference lab to do just that, to aid pathologists, really democratize pathology. I actually started a first lab in 2002 that was already established. So that was not my lab. I went to that place called US Labs, and it was very successful in transforming it, but it got so successful it got bought by LabCorp. And so the LabCorp model was not the model that we wanted, because we didn't just want to do the testing. We wanted to empower pathologists to do it themselves, to really learn about these new tests, integrate care back to the oncologists so that they didn't have everything going to the university. We wanted people treated locally. And so on the sale to LabCorp, I started a reference lab called Clariant. And the Clariant focused on cancer testing and was phenomenally successful. It really advanced all of the testing of the day, including the molecular testing, through PCR, through next generation sequencing. And it eventually got bought by GE. And then I stayed on with GE for a number of years as the chief medical officer for in vitro diagnostics, which introduced me to the combination of radiology and pathology, bringing genomics into the mix. They had an AI platform called GE Predict, and we were using that platform to understand how AI might transform. And when they closed that division, I went on and started working with Craig Venter, running oncology and immunotherapy for his startup, Human Longevity, which was all about complete whole genome sequencing, transcriptomics. We were making tumor vaccines, started doing everything under the sun.
It sounds like it had, and it's truly diverse. That makes it interesting. You kind of one that to other in some of your part. Currently, there's so many different technologies out there, especially for cancer identification, health treatments, or different parts of the country of your patient who has cancer. In your mind, and in your opinion, technologically, maybe no concept nothing and the technology. What do you think in this technology, this groundbreaker, and what do you think is in this technology will be something that's made major difference how the decisions are made by oncologists?
So there's a lot that OneCell is doing that will have impact. And I think, the one thing that's difficult to recognize for everybody is cancer is not treated by one person. So yes, at the end stage, it's treated by the oncologist, but at the early stages, there's imaging by radiologists and interventional radiologists. There's diagnostic and primary treatments by surgeons and radiation therapists. And then eventually, if the disease progresses, then you go. So you really need tools because they all have different problems, right? So some are, how can I be more confident in the diagnoses that I'm making? How could I better predict which patients need therapy after surgery versus those that maybe don't? And so, one of the things that's really unique about OneCell Diagnostics is the ability to capture CTCs live and interrogate them. So it happens at all levels. Having an AI platform that allows you to extract more value out of the diagnostic material helps the surgeons, helps the radiation oncologists. Being able to detect early recurrences of disease help the surgeon. More importantly, they help the patient. So the patient is more informed about, do they need more advanced therapy or not? Of course, if they do need more advanced therapy, then that's a prelude into them interacting with the oncologist. If their disease does progress, then it becomes even more important to have the OneCell technology where you can follow how that tumor evolves over time, because unfortunately, tumors are not static. So the more you treat them, the more you perturb them, the more they start deviating and mutating into other areas. And you need to be able to follow that. And you need complementary technologies. So we've got technologies like ctDNA, which follows the cells that are dying that we're effectively killing. And we've got CTC technology that's following the evolution of the tumor, that's showing how the tumor is evolving and predicting maybe new therapeutic options. A real-time snapshot of what's really growing. And more importantly, a snapshot of how it might have been perturbed.
Yeah. So, obviously, his technology, let's say AI is spent too. CTCs are rare, supplements in there. Then what we can't choose if it's in genomics, we could maybe that's DNA, RNA, and protein, all in the same time. And that particular targeted has it, yeah. In the next 5 to 10 years, what do you happens to this? Or, what it parties need to do in order to build and go through for testing, for solutions, for design, etc. What about in your mind? What is the relationship?
Yeah. So there's definitely some unique things in being able to capture a live CTC and being able to interrogate it, as you mentioned, even specifically for proteins, because, if we need to predict many different types of antibody drug conjugates, we need to understand what's being expressed on those tumor cells. And that's not easy to do from ctDNA by itself. You just have the DNA, they're from dying cells. You don't know what's evolving, what's being expressed, and how. So I think that one area is a separator, and that might be a true distinguisher over the next 5 years. I think one of the things that we see emerging where everybody is going to need help and they're going to need an AI platform that explains what all of these complex tests do. Because, the fact is that oncologists didn't learn this in medical school. It wasn't around during their residency. All of this stuff is evolving very, very rapidly. New drugs are hitting all of the time. And as more drugs become available, there's more options that the oncologists need to keep in mind. Because, when there's only one drug, it's easy. You give the one drug and that's it. 2 drugs, it's like, okay, I have one choice, A or B. But when there's 17 different drugs that can be given in different combinations, that's going to require help. And while there'll be guidelines, the guidelines are going to get increasingly difficult to maneuver. And if we get to true personalized medicine, the guidelines are good for categorizing people into groups, but it's not true precision medicine. It just says, oh, you've got this marker. And so, we akin that to currently a game of whack-a-mole, right? So the patient gets some abnormality and we give it a drug to knock it down, and you wait for the next one to pop up. And when that pops up, you knock that one down and you go, that's not really curing. It's a whack-a-mole game. It's just, I'm just going as fast as I can. And if 2 pop up at the same time, I don't know what to do.
So I'm more proactive rather than reactive.
So we're hoping that with the CTCs, for example, that we're going to be able to predict where that next mole might pop up and already have the hammer there so that it can't. It might not cure the patient, it might, but it will certainly delay recurrences of their tumor. And I think that's what we're really driving for, is to turn tumors into more of a chronic disease, where we can keep it at bay for 5 years, 10 years, and then it might pop up, but we'll know that it's popping up and we can intervene before there's issues. So I think in essence, what we need are there's going to be more drugs coming because they never stop. Antibody drug conjugates, bispecifics, trispecifics are sort of the next wave. I think we're going to see vaccines that are definitely going to be coming out in the next couple of years. And so that means that not that we're going to develop new tools, but we're going to develop new ways of interrogating these CTCs. And we're going to have to provide a platform that allows oncologists and other clinicians to be able to absorb the immense amount of information that we're going to be providing them.
It almost sounds like, everything you're saying seems just not just exciting, but it's like oncologists have been educated, capability, or something in the character needs to change in order for them to very quickly pick up all of this big, very high-tech, high amount of information, very quickly absorb information to be able to get action.
So I think we're seeing that now just with large language models in people's normal lives, right? You go on to ChatGPT or Grok or whatever you're using, Gemini, and you can go deep in any any topic that you want. And, we don't have those tools built specifically for oncology, but I think that's one area where OneCell has an opportunity to do. You've got these advanced diagnostic tests. You're giving so much more information than was there before. There probably needs to be, I hesitate to call it AI, but you need some way to better explain. I think the days of I execute a lab test and I give the clinician a report are going to end in the next couple of years, because what they want is just to understand what they ordered, and they would need to know how to act on what those results are, independent of, they're the ones that should say, I'm considering these 3 drugs, or what other drugs might be available to my patient based on these things. And there'll be more of them using our technology to guide the personalized care for their patients. We're not going to tell them the answer. We're going to give them the full description of everything's there, and then they need to interrogate that in the context of everything else to make better decisions. It's really about more confident decisions, better decisions, more confident decisions.
And the way to confidence really come is by better understanding. Because they have a better understanding of it, not naturally they are great in the position.
Absolutely. And it means you need all of the data. So you need the morphology data, you need the genomic data, both from the ctDNA, from the CTCs. You're going to need to know how that tumor is evolving, both from the dying compartment as well as the proliferating compartments. And so I think it's all that information going together. And as we know, they don't always align. We see things in CTCs that we don't see in ctDNA. We might not know what to do with it yet, but that's going to come out over time. The fact is there's differences, and we need to be able to identify those differences, but allow the oncologist to interact with those variances to answer the question, well, what do I do with this?
It's just a lot over here. Of course, we can talk about this, but I really want to thank you for your time. I think your insights are great. I really like what you said about, especially about 5-10 years, where it needs to go. It's not always about developing new technologies and you've been sophisticated alone. It's a lot of other things that are involved, and it's a conglomerate of all of this that you actually put together in order to make that right.
Exactly.
So thank you so much for your time.
Thank you.
Appreciate it, Dr. Glow. And
Thanks.
enjoy the rest of the time in the conference, and if there's anything else.
Thanks. Appreciate it.